Okay, here is an electron microscope image of a cell. It's actually one whole cell and parts of two others. So the entire cell, we've got most of the cell on here. It looks like we're missing just a little tip at the bottom. This is a little bit of another cell, and we can't really see a nucleus in there, so we can't do much with that. Here's a little tiny piece of a cell down here, but there's not really enough of that one that you can do much with it either, I would say. But let's get to recognize the features of a cell. So in our big cell, the one that's the entire left side of this micrograph, can you spot the nucleus? And the nucleus is huge. It's typically going to be half to a third of the volume of a cell. So really, really big. And I think you can see it. It's usually got a really dark kind of mottled outline to it, and it'll have a double membrane. If you can zoom in right here, you can see there's a little space between the outer membrane and the inner membrane right there. And right along here, there's the outer membrane, and the inner membrane is right there. So there's a nucleus. And you see this little dot in the nucleus, a little spot. That is going to be a nucleolus. So it's essentially a crystal of pure RNA that is going to go to make ribosomes. So we need a lot of ribosomes in order to keep a cell alive. You have to be expressing all kinds of things. And ribosomes fall apart. And so a cell never wants to be caught lacking. It always makes a lot of extra ribosomal RNA, and it actually just stacks it up in these nucleolus. Each of those is like a supply depot or like an ammo dump. All right, what else do we see in here? So can you spot a Golgi apparatus or more than one? I think I can. So look right up here. See these little tiny lines like that, all packed together, like little sideways pancakes? That is a Golgi apparatus for sure. And this may be another Golgi apparatus over in here. I see some little pancakiness over on this side. And it looks like we might even have a little bit going on up here. And so I would say we want to zoom into these if we can at all. I will come out of this for a sec just so we can zoom in. There we go. And there you can see all the little stacks. And yeah, don't be afraid to zoom in for sure. Here's another one. Look at all those little stacks up there. Anytime you see little pancake stacks like that, a little pile of pancakes, you know you're dealing with a Golgi apparatus. So I think we got two of them right, and the other one we whiffed on. This one might be a Golgi, but I don't really see enough there for me to definitely call it. There is certainly an amount of just experience in figuring out what one of these things is. And you just look, and whatever seems fairly reasonable, that's what we're trying to train. We're trying to train it so that we can at least get close. That we may not agree on everything, but we ought to be able to get close to an agreement. Okay. Can you see any mitochondria? Remember, mitochondria are usually going to be a little darker than everything else in the cytoplasm. And look, there's one up there. Boop, and one, and a little one, and another big one. And that's probably a baby one. There's one. You can actually see the little stripes, little slimming stripes. There's a mitochondrion. Not sure what's going on here. That might have been a mitochondrion, but it looks like it's being broken down. So that'd be kind of a 50-50 call. Let's see, I don't know if we got that one already. And that one also looks like a mitochondrion that's in some distress. Because if their little stripes don't look stripy and they're starting to balloon out, that's a sign that your mitochondrion is no longer healthy. So, Golgi apparatus, boom, there we are, and there's a nice zoomed in version. Nucleolus, bam, right there. And mitochondria, well, there's some in yellow around the edges. And there's more of more Golgis and more mitochondria rather inside the cell. But that's a pretty good number. Okay, so what do you see? What do you see? So first of all, let's look for some new features. Can you see some vesicles inside the cell? Now, vesicle is just going to look like an empty chamber, but surrounded by one membrane rather than two. So can you see them? Like up here and here and here and here and here and here. And a vesicle is typically not going to look like it has much of anything inside of it. There may be blobs coming in from the side, but that's really just the shape of the vesicle. It just wraps around these blobs. They don't have to be perfectly round. If you see a vesicle that has stuff in it, then we're probably looking at either a lysosome or a peroxisome. So there you can see that's like a vesicle, but look, it's got this weird thing inside of it. And so that actually looks a lot like a peroxisome is supposed to look. But like I was saying, we can't really be super sure just from the appearance when we're talking about lysosomes and peroxisomes. Other stuff, though, we can definitely identify. So rough ER, can you see it? I see some here. So it's going to look like a little channel, like a little path with dark stuff along the edge. And if you were to zoom in to the dark stuff, you would see that there's all little dots along the edge. Like, look at these. Look at those little dots right there. Those are individual ribosomes all along this little bit. And so this little thing is rough ER with ribosomes in it. Okay, so now let's go back and put our subtitles back on. And then mitochondria. Can you see the mitochondria? They're kind of weird shaped in this one. They're squiggling around, but they can do this sometimes. So there's 1, 2, 3, 4, 5. But look, our mitochondria, a couple of them have little vesicles inside. That one for sure, and maybe this one. That's usually a sign that the mitochondrion is not doing well. A healthy mitochondrion should be quite uniform and be covered in little stripes and should not have any open, wide open places inside of it. So these mitochondria are a little bit sick. So let's look. Vesicles, yeah, you spotted them. We all spotted them. Maybe a lysosome or peroxisome? Sure, just for illustration purposes, that's roughly what one would look like. Rough ER, I could have highlighted 50 different little stretches of it, but I picked out these couple, but there are lots more in there. And mitochondria, lastly, in orange, and I think we did get all the mitochondria in this one. Okay, now we're zoomed in a little bit closer, and you can see, look at that rough ER. Look at those little ribosomes all lined up. Oh my gosh, it's amazing. You can see the nucleus poking in here at the top, and you can see, look at these guys. You know what those are? They're mitochondria, right? And this one's got a couple little slimming stripes. This one, the stripes have started to go round, so this is like mildly sick mitochondrion. You have to basically poison these things to kill them, and sometimes the mitochondria are real sensitive and show signs of sickness first. So there's our mitochondria in red, our little big old chunk of rough ER in blue, and our nucleus with a nuclear membrane shown right up there in yellow. This cell is just a mess, but it's a cell that's actually got a broken plasma membrane, so all the cytoplasm has drained out of it. And you can see these crazy multi-membrane structures. A lysosome would look something like this, or like this, or like that, but it's hard to tell. This cell was very, very sick before it died. But what I want you to notice are all these little gray patches. See, they're kind of uniform gray. Each of those is a thing called a lipid droplet, which is just a little blob of fat, basically, inside of a cell. The mitochondria in this one look terrible as well. Like, look at this thing and look at that thing. These are very, very damaged. You see how they're really dark? They're kind of small compared to the ones in the other cells. They've got wide open holes in it, or membranes that are a mess. But if you were to zoom in on them, you would see a double membrane on the outside of all of them. And mitochondria always will have that double membrane if they're really mitochondria. So there's some lipid droplets. There's just a couple damaged mitochondria out of the many. And there's giants. Maybe they're lysosomes, maybe they're not. But just for illustration purposes, we could say that that's roughly what they would look like. Okay, can you spot the lipid droplets in here? Let's see, I'm looking. I count 5 of them. Do you see how they're just kind of in sort of a neutral light to medium gray color? Real even, yeah. And they look a little bit different from the cytoplasm. There's just nothing going on in the lipid droplets. Okay, we got that. Can you spot any mitochondria in this cell? And let's see, I see 1, 2, 3, 4. I would say 4 mitochondria. 2 of them we happen to catch long ways, and 2 of them we've cut thin ways. So they'll just look like round circles, but with the same sort of features you would find on the others. The staining is going to be a little different in every cell. It's hard to get exactly the same amount of stain in there. And so things can look a little bit darker or lighter overall, but the mitochondria are always going to be darker than the stuff around them. That's kind of how you spot them. Can you find the nucleus? I certainly see a nucleus. And inside that nucleus, I see some spots. So you could call any of those spots a nucleolus. There's not really a hard and fast definition for what nucleolus would look like in an electron microscope picture. So we can just say that, yeah, these could be a nucleolus in there. Do you see some rough ER? Let's see. Oh, yes, I see a couple of really nice bits. And I see some loose ribosomes just in the cytoplasm. And can you find the plasma membrane? That's the outer membrane of the cell. And you can see it along the top of the cell and along the bottom, and the upper right and lower left edges of the cell are cut off, so you can't see the full plasma membrane. So lipid droplets, boom, did you get them? Yeah, I bet you did. Mitochondria, well, I don't know, that one's mitochondrion, that one's a mitochondrion too, but I got 2 out of 4, so that's at least something. It's better to be sure of these things and count real stuff than to count something that doesn't really quite match. But I think these are good enough, this one and this one, that we could have counted them. They look very similar to these guys. Okay, nucleus and nucleolus. Sure, yeah, there they are, outlined in nice yellow. Rough ER, here's a couple little patches. Rough ER is usually like a long spaghetti snake, kind of like that, with 2 membranes on it. Occasionally, though, when a cell gets sick, the ER will dilate, which is like open up, like just before you have a baby. And so here's some dilated rough ER, because this cell is having problems. And then the plasma membrane. Can you see it, top and bottom? Yeah, there's the top one in purple. And if we were to trace right down here where my mouse is, that's going to be the bottom plasma membrane part that we can see. Okay, so you're going to have something kind of like this to look at. Normally we would practice these things in class, but I would say do the practice. Pause the video right now, and then let's see if you can spot the same things that I think. See if you can find at least a couple of each of these things. So lysosome, I would never ask you to do that, I think, on a test, because they're difficult to spot. I can see a thing that might be a lysosome in there. Mitochondria, yeah, I see a bunch of them, although their little stripes are looking a bit inflated. So they're just starting to get sick. They're not fully sick yet, but they don't look fully well either. Nucleus, yep, nuclear membrane, I see it. It's the edge of the nucleus, and you can actually see it's a double membrane in a couple of places. Rough ER, yes, I can see several patches of this, including a real prominent one toward the bottom of the cell. And the plasma membrane, you can see almost the incomplete plasma membrane. This cell was just a little bit of a weird shape. If it had only been perfectly round, we probably could have seen the whole thing. So, can you spot all the parts? Our lysosome would maybe be something like that, or maybe something like that. Like a vesicle with stuff inside. Mitochondria, you got them all over the place. 1, 2, 3, 4, 5, 6, 7, 8, 9. And these might actually be parts of the same one that was arching up out of the photo. And we've just cut it in such a way that it looks like a bow tie. It's a little weird. Nucleus, yep. Nucleolus, if there is one, that's the one. Rough ER is all over the place. So here's little bits of it here. Here's a bigger bit of it here. There's some little bits up here, here, all the way up at the top. It just looks like a little snake, and then there's a big prominent field of it down here, all in a row. And then the plasma membrane, yeah, it just goes around the edge. That one's pretty easy. Okay, ignore the things with stars. Practice again on this one. Pause the video, take a look, see if you can find all these parts. So, Golgi apparatus. Can you see one? Can you see more than one? I see more than one. I see one here. I see one here for sure. And I'm not sure about any others. Let's see. Mitochondria for sure. I'm seeing those. Nucleus, yeah. Nucleolus, you could say like 1, 2, 3. I don't know if you want. Counting nucleoli, it works if you're really far away from the cell. It seems like it's a real thing, but the closer you get to the cell, they're just kind of all over the place, and there's little fragments of them, and there's no good way to get an accurate number of nucleoli. Rough ER, I definitely see some. So there's a bunch of it up here. There's nice little tracts of rough ER. And there'll be some throughout the cell. I think this might be a little bit here. And there'll be little bits here toward the edge. There's a little bit of rough ER right there. Let's see. Plasma membrane, yes. And there's pretty much always going to be some rough ER visible. I would say that's a good guideline. Possible lysosome, like, I don't know, you don't know, but there's a couple of vesicles that have things inside of them, maybe. Or this thing that's maybe got some stuff inside of it. That could be, but it's hard to say. And a bit of smooth ER, possibly. Look, right up here next to the nucleus. These little single membraned blobs. That's probably going to be a little bit of smooth ER. And somewhere, three-dimensionally, this would connect up to the rough ER, but we happen to slice it and catch a slice where the smooth ER is separate from the rough ER. So there we go. That's our little practice one. And we got one more little bit of help. And then it is up to you to do this little mini project. And you're going to do the mini project individually. So everybody's going to do their own, turn it in, and yeah, do your best. You can work together, but it's got to be your opinion. And I would expect your opinion is going to differ a little bit from somebody else's opinion. And that's okay, because we'll still probably get to roughly the same place.